Hello guys, and today we will be showing you all the processes of DNA on this channel. Here we'll be showing you the roles of the ribos. First of all, let's take a look at the roles of DNA. They are the blueprint and the building blocks of life. Next is mRNA. The word messenger in the name represents what they do. They deliver DNA genetic code and turn it into ribosome. The tRNA is the key to translation because it is the key to decode the genetic code. The RNA, which then assemble the amino acids and form it into a protein chain.
So next, let's take a look at DNA replications. It's when the DNA instead of look like a spinny ladder-like structure, it becomes straight. First, there's a step called initiations. It's where an enzyme called helicase come in and separate the DNA into two. Then, the result, which is called the replication fork. Next is the elongations, which is later on add in DNA bases one by one, from 5N to 3N. But the lagging strand is different, start from 3N to 5N and start at the same time as the leading strand. There's a process called the Okazami fragment. It's a way to add DNA bases fragment by fragment instead of a nice straight line. Then the leading strand will start by a primer. Then DNA polymerase bind on top of the primer. Then the DNA polymerase, which is an enzyme, it make the DNA replication work. That is the most important part in DNA replication. Finally, there's a step called terminations, which an enzyme come in and stop the DNA replication process.
Hey guys, the next step I'm going to talk about is the DNA transcription. So, what is DNA transcription? DNA transcription is the process of copying out the DNA sequence of a gene. It happens after the DNA replication where the DNA makes a copy of itself. Transcription is the first step in a gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Transcription works with an enzyme called RNA polymerases. RNA polymerase uses a single-stranded DNA to go with a complementary strand of RNA. The RNA polymerase builds an RNA strand in the 5 to 3 direction to add new nucleotide to the 3 end of the strand. RNA polymerases link nucleotides to form an RNA strand. 1, 2, 3, 3, 2, 1. As you can see, there's 3 numbers in there, and so as transcription, because there are 3 stages in transcription. They are initiation, elongation, and termination. So, initiation happens when the RNA polymerase binds to the promoter. Each gene will have its own promoter. A promoter contains the DNA sequences that let RNA polymerase to attach to the DNA. Once the positive transcription factor has formed, then the RNA polymerase can start the transcription processes. So, for elongation, it is the stage where the RNA strands gets longer because of the addition of nucleotides. During elongation, RNA polymerase reads the template strand in the 3 to 5 direction. For each nucleotide, the RNA polymerase adds a complementary basis to the 3 end of the RNA strand. Last but not least is the termination process. It is the process where RNA polymerase will keep transcribing until it gets signal to stop. The process happens once the polymerase transcribes a sequence of DNA, also known as the terminator.
So, in all the process above, there's something in it called anticodon and codon. But both of them are different. Anticodon, which is the point where the amino acid which they beneath the tRNA, and this is an important part in translations. It used to read codon, which made from the mRNA. The codon instead, which used to read the DNA complementary nature and make mRNA. So, what is DNA complementary nature? It's TACCTTGTACCCATT, and the codon, which is AUGGAACAUGGGUAA, and that's the mRNA. Finally, the tRNA for that, which is UACCUUGUACCCAUU.
So just like, transcription, translation also has 3 parts of it, because this DNA is complex. You don't question science. Well, okay, here. So just like transcription, translation has 3 parts of it. What I like to call is stages. Those stages include initiation, elongation, and termination. So for initiation, the most important parts are the ribosome, the mRNA, and the initiator. During initiation, the ribosome assembles around the mRNA to be read, and the first tRNA, which is AUG. This can be known as the initiation complex. It is the first step of translation. The next step is elongation. Elongation is the stage where the amino acid chain gets longer. In elongation, the mRNA reads one codon at a time, and the amino acids matching each codon is added to a growing protein chain. Finally, the last stage is termination, in which the finished polypeptide chain is released. It begins when a stop codon enters the ribosome, and this is how protein is made.
So as you can tell from the process above, all of them are important to make DNA, to help it and improve it. That's it for our video. Thank you for watching. Goodbye and have a great day.
I hope you enjoyed, and make sure to subscribe, like, comment, and for more better quality video of science. Goodbye.
Exactly.